Okay, we are starting with a very good morning to all of you. My name is George Thomas. I'll explain a little bit later about what is my involvement in this. So, Professor Padayatty was my PhD guy. So, I wanted to pay homage to him in some way or the other. And I checked with Dr. Sebastian Padayatty, who is sitting here. He is the son of Professor Joseph Padayatty. And so totally agreed with me that this is the best way to do that to organize memorial lectures. Try to do it every year, once a year. So I talked to other students of Professor Padayati. They readily agreed and supported this venture. And then I discussed with Professor Parvati and Sarita Bhatt whether I can do it in QSET because Professor Padayati had long association with QSET. When this department was being started, he was the advisor who was driving it, the setting up and all that. Professor Paulos and Chandrasekharan were there at that time. And so, because of that, we chose QSAT as the venue for this event. The main idea is to inspire students. We are all science students, so to inspire science students to take up research by seeing what some accomplished scientists have done. So we try to bring different scientists who are very famous and who have accomplished much in their own right. So that is the genesis of this program. And if it is useful to all of you, we will continue to do this in one form or the other. At least one lecture a year is what we are planning. So, that said, I invite Professor Parvati to welcome here.
Good morning, and it's my honor and privilege to welcome all of you to the first lecture in Joseph D. Padayatty Memorial Lecture Series, hosted by the Department of Biotechnology and Joseph D. Padayatty Memorial Club at Cochin University of Science and Technology. This memorial lecture has been established to commemorate and celebrate the legacy of Dr. Joseph D. Padayatty, whose dedication to science and education has inspired us all. He was a passionate academician, renowned scientist, and above all, as Sir mentioned, a dedicated mentor who was instrumental in shaping the scientific ethos of this department. So when Dr. Sebastian Padayatty, son of Dr. Joseph Padayatty, and Dr. George Thomas approached us about initiating something in the name of Dr. Padayatty, we contemplated on many things, and this was one of the ways in which students would remember Dr. Padeti for a long time. So, this idea of having a lecture series is to perpetuate the spirit of scientific inquiry and curiosity, which Dr. Padeti embodied throughout his life and career. So, today we have with us an eminent speaker, Dr. K. Tangaraj, a distinguished CSAR Bhatnagar Fellow and former director of Center for DNA Fingerprinting and Diagnostics, Hyderabad. So this is the first lecture in the Joseph D. Padayati lecture series. So he is an eminent scientist and a prolific researcher in the field of population genomics, medical genomics, and evolutionary biology. He has published in our dream journals like Nature, Science, Lancet. Today, he will speak on this particular topic: who are we and what makes us unique? A tale behind tracing our roots and beyond. So, probably he'll discuss about how ancient migrations has shaped India's diversity and what our genes reveal about our ancestry, our survival, adaptation, and also health. So on the behalf of the entire Department of Biotechnology and Cochin University of Science and Technology, wholeheartedly welcome you, sir, to this program. I also welcome all the distinguished guests, faculty, staff, and students who have joined us offline and also those who have joined us online. Welcome to this program. We look forward to an intellectually stimulating and scientifically enriched session today. And I wish this memorial lecture club and the lecture series, and I hope that this lecture series will inspire us to push the boundaries of science much in the spirit of Dr. Padayatty. Thank you and welcome you all.
Thank you, Professor Parvati. I'd like to speak a little bit about Professor Padayatty, who was my mentor. I did my PhD in IISc Bangalore. So professor was working there as an assistant professor at that time. He had two projects running in his lab. One was on phage genomics and the other was on rice genomics. And when I met him and after selecting me and all that, he said, "Is it clear now?" Yes. Yeah. So many of my I think, yeah. No, there were many senior friends in IISc who advised me to take up the colitis phage work because that's easier. You can finish in 4 years' time. But unfortunately or fortunately, Professor Padayatty, when I met him, he said, "You work on rice." And then, as if he was cracking a joke, he said, "Phage is too easy to work on rice." So that's how I ended up doing rice research. The rice fellows took 6 to 7 years to finish PhD. The phage fellows would finish in 4 years and go. So I was a little depressed, but I think it was the best decision that I could have got that time. What excited me was, you take the rice seed, it's just a tissue that doesn't do anything. And at the end of the rice seed is the embryo. Major portion is endosperm. What you eat is all endosperm. By milling, the embryo goes away. If you hand dehusk it, you can see the embryo. It's a very tiny, very, very small tissue. It's just sitting there. But you give a drop of water to that tissue, and then suddenly it bursts into action. Immediately, so many things happen. It's like if you are watching a still photograph on TV and you press a button and suddenly people start dancing and singing and moving around. It's exactly like that. So, that excited me. And that is what Professor Padayatty was excited about to understand how this happens. So, my senior, Suraj Bhatt, he was one of his first students. He worked on this and he found out one interesting thing that the rice embryo has conserved messenger RNAs. That means messenger RNAs are synthesized while the embryo is being formed. They are stocked and packed and kept ready for the time when germination starts. So that is an amazing discovery in my view because the when you get water, you don't have to go transcribe any gene, all the genes required for germination, and then produce the proteins. Here, the RNA, mRNA is already ready. The moment you get water, translation starts, and so that lag time is it disappears. So that discovery came in nature, and it was a very phenomenal achievement in that sense. A PhD student getting a Nature and paper was an excellent thing. And so he and then later Iqbal Ahmed built up the foundation for rice research for me. I did some work on cloning, and Professor Padayatty is the pioneer in molecular biology and the first man, I was involved in that work, do cloning and sequencing the gene in India. So it was my work that came out like that. So we worked on histone gene organization and one thing I found that you must understand that there were no restriction in sense. There was no radioactivity available, under all these circumstances, we did that work. And I showed that histone genes, three of the histone genes I could identify, and those are transcribed from the two different strands. That means all the genes are not on code encoded by one strand, but it is distributed over two strands. So that also came in nature. So that is you know my friend and senior Suraj Bhatt would agree with me that only because we were in his lab we could achieve this distinction of having PhD work published in Nature. So what I would suggest to everybody is that keep working, keep studying, keep learning all your life. Even after you get a job, even after you retire, you should continue to do. And that's what Professor Padayatty did. Even in his 80s, he sent his DNA out for analysis. He checked up his ancestry, and he gave lectures on that. You know, Uh, when he didn't have, I don't think he was familiar with Google searches and all that, but he somehow acquired all the information and gave talks. And of course, he was an advisor here, as I told you earlier. So that's what we are trying to honor his great heritage. And one request to you people, please turn your mobile phones to silent mode. And secondly, he will be available, the speaker, Dr. Tankraj, I mean, he's a renowned scientist. I'm so happy that he agreed to come and give this talk. He will be available for about 10-15 minutes after the lecture if anybody wants to interact with him. But only one request, ask only scientific questions, not political questions. Now I request Dr. Bhatt to introduce the speaker.
Once again, a very good morning to one and all. I have the distinct honor of introducing today's speaker. I recollect meeting him a long time ago, I think more than 19 years ago, when I was training in Shitish Das lab in 2006. Long time ago. He was a very young, dynamic scientist working with Professor Lalji Singh. If I recollect, he was the director then, and also had the fortune of hearing him speak because he was one of the speakers. We had several of them during that training session. We are fortunate that we have such an eminent, erudite personality with great scholarship. I will very briefly introduce Professor Tangaraj to all of you. If you have not already Googled him, please do. I would say that he was one of the few scholars who has worked on breaking or talking down the Aryan invasion theory. But then that is another story. But let us go back to what I'm supposed to do here. Professor K. Tangaraj, born in Kanchipuram, Tamil Nadu, India, he got his PhD in genetics from the University of Madras in 1996. He started his career as a scientist B in CCMB, Hyderabad, and reached the position of chief scientist 2014. That's a very short span, okay, to become a great scholar. He was appointed as director of CDFD in 2019 and held the position till 2023. And from 2022 February onwards, he was also holding additional charge of INSTEM Bangalore. Currently, he is a CSIR Bhatnagar Fellow at CCMB, Hyderabad, and his main research interest for the last 3 decades has been in the field of population genetics, population genomics, evolutionary biology, the molecular basis of mitochondrial disorders, cardiovascular diseases, rare and recessive disorders, male infertility, sex determination, ancient DNA, forensic genetics, the list goes on. He has published more than 300 research papers, many of which are in Science, Cell, Lancet, Nature, Nature Genetics, and so on. So you can understand from the type of publication, the kind of scholarship that Professor Tangaraj holds. He's a renowned geneticist making who has made outstanding contributions towards understanding Indian population history. His study for the first time demonstrated that the Andaman tribes were the first modern humans who migrated out of Africa. It's published in Science 2005. Subsequently, he also revealed that the contemporary Indian populations descend from two groups that have mixed around 4,000 years. The two groups, ancestral South Indians and ancestral North Indians. And for the last 2000 years, almost every population in India is practicing endogamy. This was published in the American Journal of Human Genetics in 2013. Consequently, he has also shown that endogamy has led to population specific recessive diseases in South Asia. And presently, his research focus is in the area of population specific rare disorders, rare diseases. He is an elected fellow of the Indian National Science Academy, Indian Academy of Sciences, and the National Academy of Sciences. He is a recipient of several awards, including the J.C. Bose Fellowship, Sun Pharma Research Award in Medical Sciences, Raman Research Fellowship Lifetime Achievement Award, Excellence in Science Award, Distinguished Scientist Award, Sir C.V. Raman Memorial Lecture Award, Dr. U. N. Brahmachari Award, and so on. He is also the board editor of Mitochondrion, associate editor of BMC Medical Genomics, Tropical Medicine and International Health, and editorial board member of Human Genetics and Clinical Genetics. He was the president of the Indian Society of Human Genetics from 2011 to 2015, and he is the founder and president of the Society for Mitochondrial Research and Medicine. I think we have all the distinct honor of listening to such an spectacular individual with great scholarship in his area of research. So I request all of you to welcome once again Dr. Tangaraj with a great round of applause. Now invite Professor Tangaraj for the talk. Who are we and what makes us unique? A tale behind tracing our roots and beyond. Please hold on to your seats. It's going to be a very gripping talk.
So, very good morning to all of you. It's my pleasure to be here, present first Dr. Joseph Padayatty Memorial Lecture. And it's a great honor. I'm very humbled to be here. I must thank Dr. George, Dr. Sebastian, uh, Department of Biotechnology, and the Joseph Padayatty Memorial Science Club for giving me this opportunity. Truly an honor to present nearly my 3 decade of work, of course, very uh, in a way that highlight what is important for this particular audience. And as Dr. George mentioned about the scientific contribution of Dr. Padayatty and also the great mentorship. I imagine last night we were discussing that the George and his seniors published 2 papers in Nature those days. Then you can imagine how much of scientific contribution Dr. Padayatty has made, and it's truly honor to be here and make presentation. So thank Dr. Bhatt for the kind introduction. So the topic which we discussed and evolved is to know that who we are. And this kind of relative term, who we are, if you ask in the university, you can say that I am a student of this particular department. And if you go to a street, you ask, they say that they are son or daughter of this particular family and so on. But if you look at in terms of evolution, who we are and how we are all have reached here, and one has to go back and see how the human evolved, spread all over the world, reached India, and what happened after that. And the second aspect is that what makes us very unique is when you compare to the worldwide population. And this, the background will tell you that how unique we are in terms of our features, in terms of our social structure, right? In terms of every place we go, the food habit, and in terms of the genetic basis for the common disease, rare disease, also the genetic basis for metabolizing the drug, giving therapeutic response or no response or adverse response, everything all depending upon the genetic material which we all inherited over a period of time. But what happened in between and what happened very recently, that's what I'm going to cover through my intent here. So if you look at broadly that how human have evolved, although some still they don't believe on human evolution, that we human and chimpanzee have evolved from a common ancestor called hominids, right? So we share about 96% of the genome with the chimpanzee, right? So that means the 2% of the genetic mutation we opened this article saying that for nearly 80 years, evolution has been one of the basic problem in biology. It also says that Darwin and his immediate I think successors had to prove first and foremost that evolution has actually taken place. Right? I hope all of you are aware that evolution took place, and what actually he was referring is this particular book which Charles Darwin wrote, The Origin of Species. This is where he has mentioned that earlier, except human, right from orchid to whales, he has given explanation about origin of those species. But what was missing at that time was how human must have arose, evolved. But in this book, based on the anatomical similarity, he has shown that human actually evolved from the chimpanzee or apes and so on. I hope you all believe evolution. On evolution and genetics played a major role in evolution. Right? All of you believe, or is there anyone don't believe on evolution? Even if you don't believe on evolution, you can look at these three brothers who were born to a normal parents. Right? Parents were absolutely fine. But you can see they have typically like chimpanzee-like features. That means whatever the mutation which happened during the course of evolution, which changed our features, features, the way we look like today. There was back mutation which has taken back these three brothers. Right? This is one example. Second one, you can see this man and his daughter, particularly, the one of the features which we have lost during the course of evolution, the body full of air, again there's a back mutation which brought. Right? The third example, during the course of evolution, we gain ability to walk with two legs called as bipedalism. But one Turkish family somebody want to see this, the YouTube videos available. There are five daughters and one son, elder son. Only the son can walk with two legs, and also he cannot walk straight like the way we walk. He walks like a drunken fellow. But the five daughters, unfortunately, they walk with four. Again, the mutation which gave us the ability to walk with legs, there's a back mutation. Therefore, these five daughters, unfortunately, they with four. That means the genetics played a major role in our evolution, right? And the mutation, as I mentioned, mutations are the raw material for evolution. There are several genes which is already known that has helped us in gaining the way we look like today. For example, the mutation in myosin heavy chain gene is the same, could be few mutation, which created more space in the skull, therefore our brain size has increased. Again, there are several other genes have been quoted. So one of the key genes called pronorphin that involved in learning, experience of pain, perception, behavior, and memory. These are some of the unique features, right, of human. Same way, FOXP2, which is essential for normal human speech and language, some of the genes which made the jaw muscle little looser, therefore we gain the ability to speak. So these are all telling, yes, there was an evolution. And it didn't happen suddenly, the chimpanzee become human. It took about 7 million years. So during this last 7 million years, several species have evolved and most of them have vanished. For better reading, I just split it into two, 7 million years, right? And you can see the brain size of the early species chimpanzees, it was as similar as chimpanzee. And it has increased to 35% of the way the normal human brain, 50%, 60%. And interestingly, in Neanderthals, our very close relative in terms of evolution, it was predicted that brain size was slightly larger than human. We don't know they were intelligent or not. But this species is no more. A lot of work has been done. I'll come with later. But there are several interesting hypothesis has been made why this species evolved. One of the hypotheses given by Paul Miller, Cambridge, is an anthropologist. He said that inherent violent behavior of modern human probably responsible for vanishing. This could be true because we all know how violent we the human are, right? And I talked about the ability to walk with two legs, right? So that actually acquired that ability, probably got upright. Again, you can also see work with upright and so on. So slowly we evolved, and all of them have lost, including the Neanderthals. In the recent past, there's a lot of work has been done in Neanderthal. This was found for the first time in the Neander Valley, and mostly in the Europe. Neanderthal skulls were being found. So one of the scientists from Germany, Savante Pabo, he was the one actually sequenced the fossil using ancient DNA technology. It's very tough to get DNA from ancient genetic material. He successfully isolated DNA sequence and shown the missing link of the evolution between the human and non-human uh, primates, and also the other species which are shown. And because of this extensive work, he has been awarded with the Nobel Prize a couple of years ago in the field of medicine and physiology. But we know that Neanderthal and human have interbred, and therefore we all should carry some proportion of Neanderthal genome, right? In fact, in the subsequent studies, where we sequenced 300 genome, where Savanthi Peva was there, David was the one who was leading, and we were part of the study, and shown that except people in Africa, the rest are all carrying up to 3% of the Neanderthal genome. And this was further confirmed by the recent paper published by Priya Mujani, that appeared a week back, by sequencing the whole genome of 2,700 human genome. So we all carry some proportion of the Neanderthal genome, giving very strong evidence of human evolution. Okay, so this is up to the Homo sapien. Now let's look at when did the modern human arise. This is now well established that the modern human arose in Africa about 160,000 years back. And how did they migrate out of Africa? Who is the first out of Africa human? They're all questioned until very recent. We have been doing this work for last 3 decades, and to our surprise, we came to know the first out of Africa human is in Indian subcontinent. Anybody can guess who are they? So they are none other than the tribal populations of Andaman and Nicobar Islands. I don't know how many of you are aware that the typically African featured people are in Indian subcontinent, particularly in Andaman and Nicobar Islands. So we had an opportunity to go and collect the samples. That was done very long ago. And we used initially the mitochondrial DNA markers. And you all know that in the cell we have nuclear genome, the 46 chromosomes, 23 we inherit from the father, 23 from the mother. Whereas the mitochondria and mitochondrial DNA, you all inherit only from the mother. So if you want to trace the maternal lineage, use the mitochondrial DNA. So we did that, of course, very long back, sequencing the complete mitochondrial DNA. So based on the mutation exists in the mitochondrial DNA, you can actually calculate the age of the population. So what we did that about 20 years back, we have shown that the Andamanis are the first modern human who have migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. This is the first genetic evidence suggesting that the Andamanis are the first modern human who have migrated out of Africa about 65,000 years back. Right? After that, we continued our study, and human genome was sequenced, and 1 million genetic markers was available at that time for us to look at Indian population history. Then we used 1 million genetic markers. We selected 25 different population groups across India. We also used different social groups like so-called upper caste, middle caste, lower caste, tribes and primitive tribes, and all the four major linguistic groups, Dravidians, Indo-European, Austroasiatic, and Tibeto-Burman, to have a representation from across India. So in this study, what we have shown that in the prehistoric India, there were two founding populations. One, we call them as ancestral South Indians, who are part of the early human migration. That means while Andamanis were migrating, taking southern coastal route, some groups stayed in southern part of Kerala, whom we call them as ancestral South Indian. There was second wave of migration which brought people to north, and they have some kind of genetic affinities with the Europeans, whom we call as ancestral North Indians. These are the two founding populations. They have admixed and gave rise to many population groups. On the other hand, if you look at the principal component analysis, this gives you some idea about Indian populations. These are all different Indian population groups. There's a European haplog sample, Africans, and Chinese. While all of them, there are hundreds of individuals in a single cluster, suggesting that genetically not much difference among these individuals. Whereas India, if you look at every single population is unique. Although every single population is unique, but still we have some kind of genetic affinities. That is the reason this kind of gradient of clusters. Every population is unique, but still we all have some kind of genetic affinities. So what we concluded at that time was that all Indians are admixed. That means India is not inhabited by a single population. Rather, India is inhabited by a large number of small population because of the admixture followed by the endogamous. We also predict that there will be high frequency of recessive disease, for which I will come bit later. And we have refuted the Aryan invasion theory. So this comes up and goes, frequently. Some time ago, there was a paper appeared in a journal where, based on the Y chromosome haplogroup R1A1A, there was a write-up by Tony Joseph that how genetics has settled the Aryan migration, was supporting that because the haplogroup was common in Europe, Middle East, and also the northern part of India, so it was supporting kind of Aryan invasion. But later, subsequently, we have wrote again kind of rebuttal that it's too early to settle Aryan invasion theory. Of course, it clearly shows that we don't have any distinct population in India to call them as Aryan, right? There are many populations, although there are several migration happened, but there's no distinct cluster of Indian people to show that they are Aryans. In addition to the early human migration, the founding population, there are several populations which have migrated in the recent past, last few thousand years. So one example is the cities who have migrated from Africa. They are in Karnataka, they are in Gujarat. But if you look at their genome, the mitochondrial one and the Y chromosome all have admixed in the cities. The Africans and the Indian, you can see the different proportion of these genomes of admixed. This is one example. Second, the one you all know, Jewish populations are in Cochin and many other places, and these are the Jewish people, and this is called a structural analysis based on the genetic similarities of comparison. You can see they carry very small proportion of the Middle Eastern, African, and that's where the original Jewish component, but majority of them are Indian components. They're thoroughly admixed. This is the Parsis. Again, you can see this is the Indian Parsis and Pakistan Parsis. They have little admixture with Indian, but still they carry large proportion of their genome, their original genome. This is just example, and we also looked at the ancient DNA. So, you know that Harappa is the place where the modern civilization happened, and we had an opportunity to look at the DNA sequence of one of the skeleton, which was found in Harappa, particularly in a place called Rakigiri. And we found that the Indus Valley sample or Rakigiri sample consisting of some proportion of the Andamanese genome, and majority of them are Iranian hunter-gatherers, not the Iranian farmers. And this is, of course, only 300 BCE. So again, it's not majorly revealing of the human migrations, and this only shows that there is always migration and admixture has taken place in Indian subcontinent. This is again ancient DNA study, one of the largest ancient DNA study. About 500 ancient genome has been sequenced along with the contemporary sample. And here, what we have shown is that the Yamnaya pastoralites have come to India. They are the one who brought the Indo-European language. That was about 1000 to 2000 BCE, which is present in the north but not in the south. Okay, so this is again adding more complexity to Indian population group. So having all of these, this kind of hypothesis, what we have shown, is that the first out of Africa human, then subsequently there are several migrations, including Britishers, Muslim, Alexander, and so on, and these three which I showed recently. And the ancestral tribal population, something like the Andamanis or Kurumba, there are many others in Kerala. And the caste system actually evolved from this, from ancestral tribe to lower caste, middle class, upper caste. and there was again admixture took place. That's what makes India is one of the very unique place in terms of human populations. Very interestingly, some time ago, again Tony Joseph wrote a book on early Indians, where again he has described many other things. And the Indian Academy of Science had requested me to write a review on this book. We reviewed, and interestingly, what he has shown, I can zoom in further. Taking his cue from our genetic studies and other evidences, Joseph concludes that there is no pure population in India. Rather, India has mixed ancestry of separate ancestral component. Right, that's what we have shown there. Interestingly, again, he has given an example of pizza to describe this multiple level of culture. He continued to use the effective pizza metaphor for Indian population, with the first Indian as its base, with the different thickness and different region and different subsequent migration from this topic. Okay, so more than simple like this, you can't explain that how the Indian population structure exists. And very recently, Professor Balasubramanian wrote an article the last Sunday, few days back, explaining about beginning of India, and the same thing what we have shown, citing all of those. And this is a paper which is cited, and it's very clear that India is having multiple sub ancestry, found being the ancestral South Indian, then ancestral North Indian, the multiple migrations. Now I will take you to different levels to show that I showed you the admixture of many populations. So in this case, I'm giving example of admixture of the ancestral populations, ancestral South Indian and ancestral North Indians. They gave rise to many population groups. For example, you can imagine there's a group settled in a remote forest, and after finishing that food, those who are healthy, they must have migrated to another place because for food and many other things. And from there to another place. That's how many groups have evolved independently. Then these two groups have admixed and gave rise to a admixed population. This had happened last 2 to 4,000 years. The color indicates that how they have admixed. You can also take an example of our skin color, right? If you go to any other countries, you don't see such a range of skin color. No, that's very best example to show that how the admixture took place, right? If you go to any Europe, they're all light skin color. Go to Africa, the original dark skin. China, again, light skin. But in India, the only place where you can see such a huge diversity, just one example, taking the skin color alone, you can see such a huge diversity. But after admixture, for the last 2000 years, every single population maintaining the endogamy. Endogamy means marrying within the group. So now, again, another uniqueness I'm going to talk to you, that this particular uniqueness of endogamy is leading to several other issues, particularly our health issues. So that's what I'm going to talk to you now. To understand this, we took another study using 2,800 individuals from 270 population. This time we extended our study, not only restricting only in India. We took South Asian population, including Sri Lanka, Bangladesh, Pakistan, and other South Asian countries. And in this study, we've shown that it's very, very interesting and identifying a population specific mutation which might cause population specific recessive disease. I think this is very useful for the young student, that we have a lot of opportunity to study the Indian genetics. What we have shown is every single line represents one population. It's called as IBD score, identity by descent. So the more relationship in a population among those individuals, for example, if we're taking 100 individuals in two different populations, one population showed that out of 100, 80 of them are having same haplotype. That means the DNA sequence is exactly the same. That means they all have come from a common ancestor. But on the other hand, if you see only 5% of them show same haplotype, they're all different haplotype. That means there is some admixture took place in the recent past. Right? So that way, if you look at most of the populations have high IBD score when compared to Ashkenazi Jewish and Finnish population. These are the two populations well known for the founder age, but their IBD score is very tiny compared to large number of Indian. To give you an idea what it means, take the Ashkenazi Jewish. These are all the disease. These are the genes, and you can see most of them are autosomal recessive, except few autosomal dominant, one or two. So that gives that there's a strong founder event. Okay, same way the Finnish population. What it means, if you look at this pedigree, I'll explain you in a simple way. In a dominant disease, whosoever carry the mutation in one of the pairs, we have 46 chromosomes, 23 pairs. If it is a dominant disease, if it is a dominant mutation, even one of the pair carry the mutation, it obviously show the disease. But the problem with the recessive is that unless otherwise, both the copy carry the same mutation, it will not show any disease. Right? So that is the problem. If you look at this pedigree carefully, I hope that all of you are aware of the pedigree. This is a circle mean female, this is a male. If you put line, that means these two are wife and husband. And if you put line down, and these two are their daughters. If you put square further, and the son, right? This is typical example of how the recessive mutation is being transmitted. So one chromosome is normal, this chromosome carries the mutation. Because the recessive mutation, there's no disease here, right? But it is transmitted to the next generation. Again, both the daughters carry the mutation, but in heterozygous, therefore no disease. And she carried somebody from outside without that mutation, therefore again the next generation carrying the mutation heterozygous. Goes generation after generation in a next generation. But after some time, after several generations, in a village setup where 100 families are there, all the 100 must have come from a common ancestor. But if you go now, they may say that, okay, we are not related, but it's the same community. But in this case, if you see this lady or the daughter of this carry the mutation in heterozygous, absolutely normal. Again, this mutation came here. This boy carrying the mutation in heterozygous condition, again, obviously normal. But if these two marry, then there's a 25% of the chance that they contribute this abnormal chromosome and they become homozygous. Therefore, there's a disease. So that condition is very, very high among Indian populations. You can see in this IBD score, with this small, tiny IBD itself, there are 25, 30 diseases have been identified and mutation has been identified. But you can imagine with large IBD score, how much of problem in most of the Indian populations we envisage, right? So, give you example. That is one of our early studies where you can see there's a gene called polymerase gamma. It's a nuclear gene which is essential for mitochondrial DNA replication. And in this case, this is a wild type, this is a mutant, and this is a heterozygous. Right? Parents are heterozygous, as you see here, carrying both C and TLE. 45 years, 39 years, absolutely fine, no problem at all. But unfortunately, the next generation, both father and mother contributed the abnormal chromosome in 2 out of 3. Therefore, causes severe phenotype, including encephalopathy, seizures, stroke-like episode, most importantly, leading to early death. They are dying at 13 years and 10 years. Right? Fortunately, this one son is in heterozygous condition. Also, you can see brain MRI showing the lesions, and you can see these are some of the diagnostic method to diagnose the mitochondrial disease. You can see accumulation of abnormal mitochondria within this circle, and this is what it causes. This is the impact wild type 9.8 kb DNA. Because of the mutation, it introduces multiple mitochondrial DNA deletion, which leads to those kind of abnormalities. So having this information, when we studied all these 2,800 individuals, we don't have any idea whether is there any disease existing in the family or not, or in the population or not, because these samples have been collected for a population study. But we predicted that there could be a large number of population by carrying a population specific disease. So we went to go back to this some of these populations. We initially selected four different population groups from Tamil Nadu and Andhra Pradesh. Went back and we found so one of the populations in Tamil Nadu is carrying the disease called junctional earlids epidermolysis bullosa. It causes a skin rash, internal organs are all affected with multiple rashes, and also leading to congenital heart disease, and the children doesn't survive with the disease after a couple of months. Parents are absolutely fine. We sequence the exome of this family and found there's a 11 base pair deletion in a gene called LAMD3. Right? So this is the first time we identify a mutation in the gene is known, but this 11 base pair deletion is for the first time we identify. So what we need to check is whether is there any such deleted individuals are surviving or this mutation is distributed elsewhere or not. So that's where the recently completed 10,000 genome, which was recently announced by the Honorable Prime Minister on 9th January, for deposition of all these in IBDC, Indian Biological Data Center in Faridabad, which will be useful for all the researchers, including all of you. You can have access for this 10,000 genome, right? And of course, this we have earlier announced completion of 10,000 genome on 27th February last year, right? And you can see that it shows that these are the 10,000 individuals. We have Indo-European and Dravidian cluster. We have Asiatic, Tibeto-Burman. These two, the Indo-European and Dravidians have much more genetic affinities compared to Asiatic and Tibeto-Burman. And this is what we have shown that ancestral South Indian and ancestral North Indians who are admixed. You can clearly show that admixture even with the whole genome. Okay, the reason why I'm showing here is that we checked whether any of these individuals having this deletion, the 10,000 genome, none of them have that mutation. Then we went on to check in the global database consisting of 8,62,000 individuals, and we found only two individuals with heterozygous mutation. This happened to be South Asian, but we could not get precisely the name of the population. Right? But they say that broadly South Asian. But none of them, none other than these two, carry that mutation. You can see that how rare, rarest of the rare mutation it is. Okay. But again, we are not sure because we found the mutation and it could be simple association. So what is the best way to assign whether this mutation is the real cause for the disease? We need to generate animal model. When you look at the protein, this is a full length protein of 1172 amino acid. And because of the mutation, truncates and it gives only 799 amino acid. Okay. So we went on to do the CRISPR-Cas based gene editing and try to develop animal model and found with this 11 base pair deletion, the pups are dying within 2 days of the birth. They are not surviving, but still within the 2 days, we could see the scaling like the way we have seen in the patient. Then fortunately, we have also got a 45 base pair deletion, which includes this 11 base pair deletion. This is a non-friendship deletion, but actually it removes only the 15 amino acid. It's not, there's no truncation immediately. Protein synthesis goes and removes only 15 amino acid. And again, we developed a model, and the pups were fine until 4 months. Then after 4 months, you can see slowly showing up the symptoms, and this is for 5 months. And 6 months can clearly show this kind of edema in the internal organs, including there's a hypertrophy, the cardiac muscle and spleen is enlarged, the stomach becomes very small. So this is a wild type and this is a mutant. You can see spleen is enlarged, this is a hypertrophy. And we had an opportunity to look at using micro CT. You can imagine the 2 days old pups, very tiny one, and we did the micro CT. You could see this is the wild type, this is the heterozygous, and you can see homozygous deletion. There's a septal defect. You can also see the interventricular septum is so thick, and cross section also shows kind of hypertrophy. And many other organs also we have screened, and all of them are showing the abnormal. I'm not showing because of the time. And when we looked at the skin, the wild type, dermis and epidermis are tightly packed, where in the 45 base pair deletion mice, you can see the clear gap between dermis and epidermis. This is the place where the blister starts and affecting the skin. Then we wanted to develop a iPSC because the child is not surviving beyond 2 days or 2 months. So we don't have actually individual surviving with the disease. Therefore, we wanted to develop a homozygous model, taking the blood from the homozygous individual, particularly the mother, and using again iPSC technology, we knocked out 11 base pair deletion on the other chromosome, in case of heterozygous, and developed a homozygous line and tested with all the markers, pluripotency markers, they're all fine. We also used this is a heterozygous keratinocyte and this is a homozygous. You can see clear difference between the heterozygous one and the mutant. There's no proper organization, including different keratinocyte markers, and this is very intact. And in case of homozygous, not intact. We also looked at many other aspects which I am not sure. So this is for our basic understanding of the mutation, what kind of abnormality it causes. Okay, and given the evidence that yes, 11 base pair deletion is the real cause for the disease. Now we have to do something for the society, right? We can't simply observe this and leave it. And went back to the society and identified a mother who lost already 2 children because of disease, was pregnant. And we brought the mother to Chennai, collected the amniotic fluid, and tested by simple PCR whether the amniotic fluid carrying heterozygous or homozygous. And we found that heterozygous band were very weak. But we confirmed by sequencing. You can see up to here is a clear, after that there's an overlapping. That means that shows that there's a deletion, therefore there's an overlap. Mother sequence, amniotic fluid, clearly demonstrating that there is a heterozygous. So we advise the mother to continue with the pregnancy. It's not like the previous two pregnancy. This child is going to go on the normal features. And it's our joy to see the child born with uh, the healthy phenotype and she's growing all right. And now we want to take it to the next step because we know the frequency of that mutation is quite high. We screened initially few individuals and found the frequency of this mutation in heterozygous is about 10%, right? But we wanted to screen for the entire village, there are about 5,000 individuals. Now we can do, we could have done ourselves, but we approached uh, Tamil Nadu government, the Directorate of Public Health and Preventive Medicine. And it took about 1 year because they have a lot of procedures. And only recently they gave an approval for us to go and collect the samples. Now we go and collect the sample. We have very systematic plan that with the help of other organizations, PHCs, Dixon, Bangalore, the local leaders and so on. And initially, we have made awareness program. In fact, tomorrow I'm going there with my team to collect the samples. Right. So from here tonight I'll go and collect the sample from there. So initially, what we will do, we collect the blood sample and saliva for all the individuals to identify the heterozygous individuals and put them in the database. And if there is any mother with pregnancy, we collect the amniotic fluid and do the sequencing. And if I identify, then we do the counseling and they can decide whether to continue with the pregnancy or not. And this database will help for the future generation. Okay, for premarital counseling, for example, children with mutations are identified, but after they grow, maybe after 10 years or 15 years, then they can take out the database and see uh, whether the boy identify the girl or girl identify the boy, whether both of them are carrying the heterozygous mutation or not. In that case, we can advise them. They can still marry, but they have to do prenatal diagnosis to see whether the fetus carrying the mutation or not. And if they are one is heterozygous, one is wild type, no problem. A number of child also will carry the most heterozygous. So that kind of uh, database and counseling is the one which we are planning now. If you do this for next few generations, in fact, we can eliminate the mutation from the community itself. Therefore, they don't have to really worry for this. So the next generation will be absolutely fine. So that's what is our recent research focus is taking the genetic to the society. It's not this one. We identified several of them already, and you can see that the Kashmir families and a lot of families in community have that. So having this information, when I was in CDFD, I initiated a program called Pediatric Rare Disease Genetic Disorders, where 16 institutes across India who are doing genetic work are part of this program. They do initial screening, then send the sample. If they don't find any mutation to CDFD, CDFD will do either exome sequencing or whole genome sequencing. And the process is same as what I explained. Initially, we predict the mutation, use one of these animal models to see whether, because mostly what we expect is the normal mutation. And if we see the similar phenotype, then we try to use lentiviral based therapy, particularly for hematological disorders. Otherwise, do very strict counseling procedure. And most importantly, we want to make awareness program. And in this particular website, somebody is interested, they go into the website. There are videos on 8 different languages for the common people to go through. And anybody can go through and get back to CDFD or any other institute. In Kerala, Trivandrum, RGCB is also part of this. So through this, we can achieve a lot. And in fact, the former Vice President Sri Venkaiah Naidu has inaugurated Pediatric Rare Genetic Disease Laboratory. We have found full name for that. And Secretary DPT Dr. Rajesh Gokhale has inaugurated, launched this particular program, and going on very well. So with this, I'll stop, and I will be really happy if any of you have any question. And as the judge said, you can ask any question, but I can avoid if it is political. And before finishing, I would like to thank all of those who have contributed. It's not that only my laboratory alone worked on that. There are several people who have contributed across not only India, across the world. You can see some of them are outside India. And majority of the funding has come from all government institute, the Genome India and Pediatric Rare Disease, but both I am still the coordinator, come from DBT, CSR, UKRI, DSP, and Medical Research Council program just for over. So I must thank all of this, and thank you again for listening talk. Once again, I thank Dr. George, Dr. Agustin, Department of Biotechnology, and Memorial Lectures Club, and all of you attending this talk. Thank you. Yes, yes, please. Second slide.
Ah, yes. So you have been looking on a very like entities, but we're looking at it with a microscope backwards, starting with I don't know what is that the peak of the
So that's where the divergence
okay, I don't know what's the
took place.
peak. Anyway, that's
There's
not
no peak. That means this is from a common ancestor. For example, if you look at the hominids, from hominids there is a divergence.
can I
Chimpanzee
check
and
what
human.
is the upward the top?
This
What is what happens? What is there?
So that's where there's a split. For example, if you, I know very well that the human and chimpanzee split took place about 7 million years. Similarly, this could be several hundreds of million years. Yeah.
What are
We can say there's a divergent time, species of divergence.
this? Now, if you try to look at from 1,000 years later sitting in the same room, based on your study for so many decades, how do you think this slide will change?
So this won't mostly change. 1,000 years, for example, the evolution is taking place at micro level. Even if you take human, and for the last maybe 10,000 years, there's a lot of mutation arose because of the environmental influence. For example, our skin color about 5,000 years back, or 7, 8,000 years back, were black, and people migrated to Europe, and they could not actually synthesize vitamin D with a very mild light, and therefore they acquired a mutation in SLC24 5A which turned the skin color into lighter because the melanin content is low. So that we acquired. Similarly, the lactate persistence gene mutation after animal domestication, after drinking the milk, that mutation arose about 5,000 years back. Okay, so even the sickle cell mutation because of malaria, so slowly we are gaining those mutations. It's not going to give a new species because we acquire this mutation, but at the species level, it's not going to give much within this 10,000 years because it takes millions of years.
So the building is full some point in time.
So at this point of time, it's very difficult because anything abnormal, somebody 10 feet height, suddenly it happens. For example, three brothers who acquired chimpanzee-like feature. and nobody is going to marry them, and therefore that gene is going to go to the next generation. That's the end of it. Similarly, it's a normal totally. That's the end of that mutation. It's not going to go to the next generation.
No, sir.
Simply, that's why we cannot predict the evolution. We may gain a lot of information because we have seen in the last 2 generations, we ourselves, a lot of other things have changed, right? So that kind of changes happens, might give us more knowledge, give us more information, but it cannot produce a new species within short span of time. I don't know if you got it or not.
Why do you know? Yeah. Yeah. So I want
So we can look at the past because of the evidence, because they're all evidence based. But we don't have any evidence to predict because it can go in any direction. So future is very difficult to predict. So even there are in the news, yeah, there's the news that the Y chromosome is shrinking, and they say that the male species is going to vanish. Right, right. It's not going to happen. Right.
it's yeah, students please ask some question. It was meant for you. Why did you choose this map for me?
That is mainly because of the culture. Fancy has like this. We will change it up or something. Yeah, any students can ask. I guess yes.
Early, uh, so they have my
We don't have any evidence that similar people have lived there. They must have just keep walking. They might not have seen a suitable place for them to live there. So they were moving. Unfortunately, we don't have any archaeological site to excavate and find is there any such thing exists. But we have further, they did not settle only there. Some of them migrated to Australia. Even the Australian Aboriginals and the Malays are still sharing some kind of genetic code.
they had to
Yes, yes. They have the ability to make small boats. They cut the tree, dug out that we predicted based on the genetic evidence.
happen from. Can it happen because they were confident over and our border?
Now you are asking. That's also not everybody. Even the Great Andamanis have admixed with the mainland Indians. We looked at the Y chromosome. Only four of them, because the total number is reduced to 20. Only four males we look at. Each Y chromosome is different. The original Y is not there. They are mixed. On the other hand, the Onge and Jarvas carrying the original Y chromosome.
And it can be harmful. Uh,
There's only one sample. We found that maybe around 65% of Iranian hunter-gatherer, which we predicted about 10,000 years. But the sample age itself is not that old. It's only 3,000 BC. You can sit and Yeah, they have been brought by Portuguese traders and they sold to Nawab and Sultans to work in their army. Yes, yes. Some of them, but most of them are mixed. Some mutation, uh, uh, there's they don't have sickness, but we look at G6PD for the same purpose, malaria resistant. In fact, they have lost some of them because of the admixture. So we generally see about 60% of them carrying the mutation, which gives them advantage over the malaria. Whereas now they have only 10% because of admixture. Similarly, the Jews, they have advantage. They have more of cancer-specific mutation. Now that has reduced because of the admixture. Some place they gained advantage of admixture. Some place they have lost.
What about butchers in Kashmir? They are the sheep rearing. They're shepherds.
Yeah, yeah.
Anything special?
We analyze that
anything-
nature genetics paper. They have their IBD score is much longer. That means they may have more of population specific mutation.
I saw in that graph.
Yeah, Gujar was there. Very long. Yes. Students, no, no questions. You understood at least some part.
Students have what is the polio and COVID virus, coronavirus? The genetic sequencing and you on that anything interesting to ask. See if a polio case in West Pakistan is from this and all. You keep hearing always it happening. I think biotechnology students should be more alert or also find virus. Maybe we are trying so I like one. Oh, yeah. One was not happening.
So, in fact, I showed human and chimpanzee, sorry, human and Neanderthal can happen, right? Can happen, right? But
One.
for no selection pressure through the environment, for example, malaria is one pressure. High altitude is another pressure, where we need to carry more oxygen because less oxygen. So the RBC had the ability because of the mutation to carry more oxygen. So these are the smaller micro level evolution. It's not going to change the our phenotype or any other major aspect. Again, so that happened. The children are not surviving. No, that's end of that. But because the heterozygous condition doesn't affect the phenotype, it gets spread because of purely because it's a recessive nature, right? Except for homozygous, which gives the phenotype. Otherwise, no other issue in the heterozygous condition. That's why it's carrying on for the last several hundreds or thousands of years. Yeah, it will remain. Yes. If it is a dominant, probably would have lost because it's not disadvantage in the heterozygous condition. And it's continuing. And because of the endogamy marriage practice, there's also one point which I didn't mention is that the same population is in 5 different districts, but other four district doesn't have this. Only one district. That means within the same community, they're not going out of this district to other districts. Such a huge substructure existing in that population itself. Gene therapy will work when only when, for example, there's a late onset disease. For example, cardiomyopathy, it affects after 35, 40 years, and people have tried, but it's not very effectively used. But here, it's a lethal, right? And rather than doing all this, avoiding is the best for any genetic disease.
I think gene therapy and there are stats of a few early coming together. Mostly, you avoid such marriages, and that's what happening in sickle cell counseling now. Yeah, no, I'm just wondering, this scenario that in earlier case there was no human intervention, and now there's human intervention in selection of entities. So over time, how this will pan out? It's me. I'm just
That's what the future. Somebody really wanted genetic to be major player in our future perspective, then one can sequence the genome and predict the mutations, particularly which is recessive in nature. Same mutation, you can look for that in the opponent, no, the or before marriage. You can look at and do the selection, but still, that alone may not give you. There could be something happened at the no, early embryonic stage that might also create some problem. But to some extent, you can avoid. People are using it. So you're talking about the Neanderthal. Right? So there could have been competence between Neanderthal and human. So although they admit they married the Neanderthal, but that was located only in the Neanderthal Valley and the Europe. Probably by then, human must have reached other continent, and their population size is much bigger than the Neanderthals, right? Although they would have been intelligent because of great size and so on, but the population size would have been very small, and that's the reason probably, as Paul Muller predicted, the violent behavior of the human would have, you know, led to no vanishing or killing of all the Neanderthals. That's one possible.
Also, humans cruelty.
Uh,
That's probably another fact that eliminated.
and that's why I'm saying.
services.
Thank you, Dr. Tankaraj, for this wonderful talk. I think you must have gotten a good idea of how basic research can lead to great applications which are of tremendous value for human beings. So I urge you to follow that. Basic research is good, and eventually it will find an application for human research. So now I request Sebastian Padayatty to propose a vote of thanks and then give a memento to our speaker.
I'll be very brief. Let me thank Professor Parvati and Professor Bhatt for hosting this conference. And this would not have happened without George Thomas putting in a lot of effort. He's the one who's conceptualized this talk. Uh, to to go back to just one point, to go back to Dr. Kankaraj's focus on endogamous growth. We in India have probably 5,000, 3 to 5,000 endogamous groups who are bred true for 1,000 years or 2,000 years. We can look upon this as an experiment of nature, like mice which are purebred mice, which are used in the laboratory. And he investigated rare diseases. And an example where you have a very common disease is Pima Indians in the southern United States. Some of them live in Arizona, some of them live in Mexico. In 1930s, Dr. Jocelyn, who founded the Jocelyn Diabetic Clinic, which is a preeminent diabetic center in the world, he lived in among Pima Indians for 3 months. He could not find a single person with diabetes. In 1990, 50% of Pima Indians have had diabetes. And it is even higher now, probably 60, 70, almost 100% of them are obese. So in 60 to 70 years, a single endogamous group went from 0% diabetes to the vast majority being diabetic and with renal complications. To the extent that the National Institute of Health in Washington has its only branch uh, dedicated to studying the single endogamous group. Now, if we were to study our endogamous groups, we will need not one Tangaraj, but 5,000 Tangarajs at hundreds of institutions. Uh, but to look at it in another way, this experiment of nature is a national asset. And by studying this in detail, we do not know what kind of things we are going to find and what kind of magical remedies we can devise in the future. So it is a tremendous start, but we have a long way to go, and we all have to thank Dr. Kangaraj for this outstanding work over many decades. And being kind enough to come down to Kochi from his busy schedule and give this talk. So, thank you very much.
Sir, you didn't want to speak in public but want to ask it privately, now is the time. You have a few minutes.